let's get into it. Let me just grab a quick sip. So inquiry question is, how does mutation introduce new alleles into the population? So module 6 is a really nice segue from module 5, where now when we look at mutations, firstly we're going back to our processes of replication, translation, transcription, and talking about mutations affecting those processes. Okay, here we are. So first question we have here is that we're going to look at is, how do mutagens operate, the different types of mutations. We're going to talk about somatic versus germline mutation, and then talk about the effects of mutations on coding versus non-coding DNA, and then look at causes of genetic variation, and finish off by talking about population genetics. So we're going to talk about mutations, gene flow, and genetic drift. So here we are. Inquiry question is, how does mutation introduce new alleles into a population? Remember, a mutation, firstly, is a permanent alteration to the nucleotide sequence of an organism's genome. This is where we are permanently altering someone's or rather natural circumstances or their own system leads to the alteration of someone's own nucleotide sequences, which then changes the proteins that they are able to make. Awesome. Now let's have a look, and I know it's a long dot point, but I believe this is one of the last parts of this dot point. So let's have a look at the effects of mutations, gene flow, and genetic drift on the gene pool of populations. So here we are. So a mutation, remember, is a permanent alteration to the nucleotide sequence of an organism's genome. It leads to the creation of new alleles. It can be favorable, and it can also, it may be favorable and subsequently selected. Gene flow is the transfer of genetic variation from one population to another, leads to changes in the frequency of alleles, and it can affect the distribution of genetic diversity. And finally, genetic drift is when the frequency of alleles within a population changes because of either disappearance of particular genes, and that could be a result of natural selection over time. So for example, traits can become more or less prominent, and it can also lead to evolution of species and creation of new species as well. So when we talk about genetic drift, let's just talk about a couple of causes. So that, and this is kind of like a bit of revision from year 11. So bottlenecking is a process in which there is a abrupt reduction in the number of individuals. It could be due to natural causes, a sudden severe selection pressure, and that leads to a loss of diversity. The founder effect, of course, is when a new population is established by a small number of individuals. So they separate from the main population, they establish a smaller population, and it leads to a loss of genetic variation within the new group, and it can also lead to new speciation events as well. Evolution, overall, remember, is the change in a species over many generations. It results, and as a result of natural selection of favorable characteristics. And remember, when we link this back to evolution, it's because we're talking about mutations, how it leads to genetic diversity, and how it can then lead to these specific processes. Remember, variation occurs within a population due to natural selection. My bad, right there, natural selection. Environmental pressure is applied to population, and phenotypes best suited to the environment, to the changed environment, will survive, and surviving organisms will reproduce. So this is telling us that genetic variation is super important, and I'm trying to draw a straight line, but that's not working. So genetic variation, GV, really, really important. And I want you to remember this, because we're going to come back and link this back to biotechnologies and genetic technologies when we